Hello all NEET and EAP set aspirants, this is your Prasanna ma'am from NEET World. Welcome you all for the most interactive and interesting session. Now we are going to solve all the match the following type of questions from the chapter molecular basis of inheritance. Without much delay, let's get started. The first question, where this question is from the gene expression or Lac-Operon model, where different sites are given and the activity at that site is provided in column 2. If you look into the Lac-Operon model keenly, Lac-Operon model, where initially the promoter site is there, promoter region is there, then operator region, which is followed by structural genes Z by A, and lastly it is terminator, and this is from 5 prime to 3 prime. The promoter region, which is at the 5 prime end, to the promoter region RNA polymerase will come and bind. What will bind beta? RNA polymerase will come and bind. To the operator region, repressor will come and bind. To the operator region, what will bind? Repressor will come and bind, and it will repress or it will restrict the process of transcription. Now if you look into operator site is the binding site for RNA polymerase, this is option 1. It is the binding site for repressor. Operator site is the binding site for what ma? Repressor, this is option 2. Promoter site is the binding site for RNA polymerase, this is option 1. Regulator gene which is far away from the operator. Here in the Lac-Operon model, regulator gene, which is I gene, regulator gene is what ma? I gene, it will synthesize repressor. It will synthesize what? Repressor. So it codes for repressor, option 4. Structural gene will code for either protein or enzyme. If you look into Z gene codes for beta galactosidase, Y for permease, and A for transacetylase. So yes, D is option 3. If you look into the options carefully, A is 2, B 1, C 4 and D 3. Option 2 is the correct answer. Next, match the following type of question again. Here column 1 different regions are provided and 2 their activity is also provided. Exon means expressed sequence. I said this is the way you can remember it easily. Exon expressed sequence or coding sequence, so it is option 1. Intron unexpressed or inactive sequence which is non-coding, here it is option 2. Next, genetic code is a triplet codon, right? It triplet basis will be there, triplet codon on mRNA, it is option 3. DNA packaging, the very much well established or accepted model of DNA packaging is nucleosome model. What is it ma? Nucleosome model, where it has histone octamer. What does it has? Histone octamer, which has 2 sets of H2A, H2B, H3 and H4 proteins. It has 2 sets of H2A, H2B, H3 and H4 proteins. These positively charged proteins are filled with amino acids lysine and arginine. These are positively charged because of basic amino acids, that is lysine and arginine. DNA, which is in the chromatin form, is wrapped along this histone octamer and they get condensed. Okay, and DNA is linked from one histone octamer to the other through a linker DNA which has H1 histone proteins. It is linked from one octamer to other by linker DNA which holds the H1 histone proteins. This is a gist of nucleosome model. So this you have to remember only one thing. Nucleosome model has histone octamer and these are the proteins, histone proteins which are involved. They are positively charged because of the amino acids lysine and arginine. Okay, so yes, this is how DNA chromatin material is packed. So DNA packaging means nucleosome, option 4. So yes, exon expressed sequence, that is 1, A is 1, B is 2, C is 3 and D is 4. Option 3 is the correct answer. Next question, if you look into here, in column 1 RNA polymerase is given, and in column 2 what it is synthesizing is asked. Always fix it in your mind. Many a times the question is asked from RNA polymerase. RNA polymerase 1 will synthesize the structural RNA, that is rRNA. Okay. RNA polymerase 2 will synthesize mRNA or messenger RNA. RNA polymerase 3 will synthesize tRNA, which is also called as soluble RNA. So, if you look into the options clearly, A is 1, B is 3 and C is 2. Option 1 is the correct answer. Next question, if you look into, here if you see, again enzymes are given, their activity is provided. Beta galactosidase will cleave or it will break the disaccharide, that is lactose, into, lactose is breaked into galactose and glucose. Lactose is breaked or hydrolyzed into galactose and glucose. So, A is option 3. Next, permease. Permease will increase the permeability of beta lactose into the cell. Okay. Permease is coded by the gene Y. Beta galactosidase is coded by the gene Z. Okay. So, B is option 4. Ligase will bind all the DNA fragments. They will join the DNA fragments. Here it is option 1. Ribozyme will play catalytic role in joining the amino acids. Ribozyme will play catalytic role in joining the amino acids, and it will release an enzyme, that is peptidyl transferase. It is what ma? Peptidyl transferase. Okay. So, this is the enzyme that ribozyme will provide. It will join the amino acids and it will make the peptide bond. So, here it is option 2. If you look into the options cleanly, A is 3, B 4, C 1 and D 2, it is option 2 is the correct answer. Next question, if you look into beta, here the genetic code is provided and their amino acid, what is coding, that is given. Mostly genetic codes are not asked in your exam, but basic codes you have to remember ma. One is AUG, this is called as starting codon. This is called as starting codon, or it also codes for methionine, so it is option 2. And you know stop codons or terminating codons, right? You have to remember this for sure. UAA, UAG and UGA. These are the stop codons or terminating codons. Next, UUU is coding for phenylalanine. Here it is option 1. B. These are stop codons. These are facilitating the process termination. Next, UGG. This is tryptophan. These three though you have to remember. Most importantly, start codons and stop codons should be in your mind. So, if you look into the options, A is 2, B 4, C 1 and D 3, option 1 is the correct answer. Next question beta. So, here match column 1 and column 2 again. Here the activity is given, we have to choose with respect to column 2. Splicing means removal of introns. Splicing is a post-transcriptional event. Splicing is what ma? Post-transcriptional event which happens inside the nucleus of eukaryotic cells, wherein in splicing we will remove the introns which are non-coding or inactive sequences. So, 1 is option D. Okazaki fragments are lagging strands. Okazaki fragments are what beta? Lagging strands. We have learnt in DNA replication, no? Yes. Jacob and Monod has proposed Lacoperon model, which is option A. Next, inducer is what? Inducer in the Lacoperon model, it is lactose. Inducer in Lacoperon model is lactose, that is C. So, if you look into the options, 1 is a D, 2B, 3A and 4C. Option 1 is the correct answer. Next question beta, next question. mRNA will provide the codon. mRNA will provide the codon for translation. mRNA will provide the codon for translation. mRNA from 5' to 3' starting from start codon it provides, okay, starting from start codon, it will provide the codons for transcription, and tRNA will copy this with an anticodon. tRNA will provide what beta? An anticodon, and it will copy this. tRNA will give an anticodon. mRNA will provide codon. Okay, this you keep in your mind during the process of translation. So yes, mRNA is providing codon. B, anticodon is provided by tRNA. Option A, semi-conservative method of DNA replication is experimentally proved on a prokaryotic cell, that is E. coli. E. coli is the model organism, you have to remember, and yes, this is option D. Griffith gave us transforming principle on working out on the bacteria Streptococcus pneumonia. Griffiths gave us transformation of bacteria, that is option C. So if you look into it properly, 1 is B, 2A, 3D and 4C, option 2 is the correct answer. Next question beta. Yes, match column 1 and column 2 again. Here 5 prime to 3 prime AUG is given. What is AUG ma? AUG is an initiating codon. So here it is option D. RNA with introns and exons is called as hnRNA, which is subjected to post-transcriptional events. Gene is a segment of DNA, which is functional unit of inheritance. Gene is what ma? It is a segment of DNA. This is the DNA, sorry. If this is the DNA, gene is a segment on DNA. Yes, and gene has 2 pairs of alleles. Okay. Nucleosome, just now we have discussed about nucleosome also. It will condense the chromatin, or it will pack the chromatin. It is option B. So 1D, 2C, 3A and 4B. Here it is option 3. Next, termination. Termination during the process of translation. Always do remember, initiation is facilitated by initiating factors, termination is facilitated by release factors. So yes, termination is facilitated by release factors, that is option 3, GTP dependent release factors. Translation, the very first step of translation is amino acyl tRNA synthetase. Here it is option 1. Transcription, transcription is catalyzed or promoted by RNA polymerase, that is option 4, RNA polymerase 2. And DNA replication, you will find Okazaki fragments in the lagging strand. Here it is option 2. So if you look into the options clearly, A3, B1, C4 and D2, option 1 is the correct answer. Next, scientists is given and their activity is provided. Griffith gave us the transforming principle, that is option D. Hershey and Chase transduction, option C. Prokaryotic DNA, we will call it as nucleoid A. Euchromatin is an active chromatin. B. Heterochromatin is inactive chromatin, which is densely packed. So here if you see 1D, 2C, 3A and 4B, option 3 is the correct answer. Next, Frederick Mister is the first person who analyzed the DNA, but he named it as nuclein. This is option 2. Griffith gave us transforming principle working out on Streptococcus pneumonia. This is option 3. Next, Hershey and Chase worked on bacteriophage, which is option 4. Okay. Watson and Crick gave us double helix of DNA, which is option 1. Wilkins and Franklin worked out the double helical model of DNA through X-ray diffraction studies, which is option 5. Okay. Now if you see clearly, A is option 2, B3, C4, D1 and E5, option 3 is the correct answer. Next question, if you look into DNA digesting enzymes. This you will come across the biochemical process of Avery-McLeod, okay, Avery-McLeod, those biochemical process. Here you need to look into the enzymes clearly when you are going through that experiment. Remember properly, DNA digesting enzymes are called as RNA digesting enzymes called as RNAs, protein digesting enzymes protease, DNA digesting enzymes DNAs, fat digesting enzymes or lipid digesting enzymes is lipase. So A is 4, B 3, C 2 and D 1. Option 3 is the correct answer. Next, sigma factor, it will promote initiation. You have to remember all the time initiation and termination of transcription process. The process, entire transcription is facilitated by RNA polymerase. We know RNA polymerase along with sigma factor which is said to be a holoenzyme. Along with sigma factor, it is said to be what ma? A holoenzyme which will promote initiation. This will promote initiation. Okay. RNA polymerase along with Rho factor will promote termination of transcription. Okay. Sigma factor, initiation. It is promoting what? Initiation when it is binding with RNA polymerase. Capping is done at 5' end. Here it is 3. Tailing is done at 3' end of hnRNA. Okay. Both these are post-transcriptional events. Both these are post-transcriptional events which happen inside the nucleus. Both 5' capping and 3' tailing occurs in hnRNA. Occurs to what ma? hnRNA. Next, coding strand is from 5' to 3'. Here it is option 1. So A2, B3, C4 and D1. Option 2 is the correct answer. Next question, if you look into, here bacteriophage lambda has these many nucleotides, 48,502. So here it is option 1. Human being, diploid cell of a human being or a diploid human cell will have 6.6 into 10 to the power of 9 base pairs. Option 3. E. coli has 4.6 into 10 to the power of 6 base pairs. So here it is option 2. So A is 1, B3 and C2. This is from the first table of your chapter molecular basis beta. That table is very much important. Keep it in your mind. Okay. Taylor and his colleagues, they gave us the experimental proof of semi-conservative DNA replication in eukaryotic cell. Michelson and Stahl gave the experimental proof of semi-conservative mode of DNA replication in a prokaryotic cell E. coli. Taylor and his colleagues gave on eukaryotic cell. So A is 4. Next, Frederick Mister, we have discussed, he gave us the term nuclein, option 2. Crick, Watson and Crick, they gave us central dogma of molecular biology, that is replication to transcription to translation. Okay, where DNA is replicated and information is passed to mRNA, and then from the ultimate goal of DNA is to synthesize a protein. Okay, so yes, Crick is central dogma 3. Next, Avery-McLeod MC Carte gave the biochemical characterization of transforming principle. Just recently we have solved that objective, where you have to be clear with RNAs, protease, DNAs. What is their activity? Okay, so D is option 1. A is 4, B2, C3 and D1. Option 1 is the correct answer. Next question. rRNA is the, just a second, rRNA is doing the catalytic role during translation. mRNA, as we have studied, it is providing the template, it provides the template. We have observed in the previous question, right? mRNA will provide the template codon, okay, during translation. So this is 4. rho factor is a termination factor 1, sigma factor is an initiation factor, which is option 5. tRNA, which is soluble RNA, this reads the genetic codon and tRNA will provide an anticodon. This is option 3. So yes, A2, B4, C1, D3 and E5. Option 1 is the correct answer. Next question, if you look into beta, yeah. LAC-Z is synthesizing beta galactosidase. LAC-Z is synthesizing what ma? Beta galactosidase, which is option 3. LAC-A will synthesize trans-acetylase. LAC-A will synthesize what? Trans-acetylase, which will transfer acetyl group. This will transfer what? Acetyl group. So it is option 2. I gene will synthesize repressor. I gene will synthesize repressor, which is option 1, and operator is the attachment site or binding site of repressor. Once again, for your idea, let me provide this mRNA chain, okay, from 5 prime to 3 prime. First promoter region is there, then I gene, okay, as it is provided here I am writing I gene, which is again followed by a promoter, operator, then structural genes ZYA, lastly it is terminator. To the promoter region always RNA polymerase will come and bind, which will start the reaction, and here regulatory region or I gene will release out the repressor. Okay, again at the promoter region RNA polymerase will bind. If this repressor is free in the cytoplasm, this repressor will come and bind to the operator region. It will stop the process of transcription. It will stop the expression. It will stop the gene expression. If lactose, which is inducer, if it is present, if lactose, which is inducer, is present, lactose will come and bind to this repressor region. Lactose will come and bind to this repressor region. Now repressor cannot bind to the operator. Now the gene expression starts. Structural gene Z will code for beta galactosidase. It will cleave, beta galactosidase will cleave lactose into glucose and galactose. Y gene will synthesize permease, which will increase the permeability of lactose into the cell. A gene will synthesize trans-acetylase. This trans-acetylase will transfer the acetyl group. Okay, it will do what? It will transfer the acetyl group. So yes, it is option 4. Clear with the gene expression? I have dealt this very shortly because you can remember this or recollect it very fastly to answer this question. Okay. So yes, A is 3, B is 2, A is 3, B is 2, C is 1 and D is 4. Option 4 is the correct answer. So yes, before ending the session, I would like to tell you about this news that in NEET World, which is located in Narayangoda, Hyderabad, we are going to start a crash course, which is a 40 days program where you will be having daily practice tests, weekly part tests and 10 mock tests for free, which you can access both in online and offline platform. Crash course is also provided both in online and offline platform. If you want to take coaching in offline platform, including hostel fees, it is just 15,000 beta. You will get a very much quality coaching here. We ensure that you are getting very much quality coaching, and stress-free education is our agenda. So this is golden opportunity to all the students who have completed 12th standard and 11th standard completed students. You also can join. 11th class completed students have free crash course in our institute, which is offline. You can contact to this number for further details. Yes, thank you.